Hello, welcome to another screencast where this time we'll be continuing discussion of signal transduction pathways. And this time we're going to be describing and explaining here variation in the receptors. Right? So we're going to be talking about G protein linked receptors and tyrosine kinase receptors. We're going to focus on these two, but then we'll mention ligand-gated ion channels as well as intracellular ones, okay? And then we'll handle the elements of the transduction process in another screencast here. All right, so let's get started here. So the big two, the G protein linked receptors, right, here are a class of receptors so that are associated with a G protein. And so the image at the top here in A, right? So this is in the inactive form. So we are not, we haven't signaled any sort of response yet. So here is the G protein linked receptor, and it's linked to a G protein, right? So this is the receptor, right? Here is the G protein, right? And you notice that in an inactive state here, it has GDP bound to it, right? Now just real quickly over to the side here, right? GDP sounds like ADP, right? So then we're going to be using GTP, right, which sounds like ATP here. Right? Now G and A are both purines here, and so it's not surprising that GTP and ATP have similar functions here. So this is why this is a G protein. Right? Then the third component of the mechanism is over here, this enzyme right over here, which is often adenylyl cyclase. Adenylyl cyclase. Now, so these components here, the G protein linked receptor, the G protein itself, and this adenylyl cyclase. So this is what happens with a G protein, is that the signal molecule is going to bind to the activated receptor, or sorry, to the receptor. This activates the receptor. That in some way is going to trigger the G protein to release the GDP and then bind GTP. And so now this GTP bound to the G protein is an active G protein. This active G protein actually shuttles and moves its way through the cytoplasm here a little bit and will now bind to the inactive adenylyl cyclase. Right? And this binding right here, right, activates the adenylyl cyclase. Right? So this is what we're saying, this is activated enzyme. That activated enzyme then sets off a series of reactions, right, involving different enzymes. Right? And then that ultimately is going to lead to some sort of cellular response here. Right? So there's an awful lot of steps there that you should pause and sort of go back and take a look at, right, and review. When you want to stop this, of course, the way to do that is you remove the signal molecule. Right? So this is either removed actively, removed, or it's oftentimes degraded. Right? And so if it's degraded, that means it's, in some reason it's in low concentration. Right? Here the signal molecule is probably in high concentration. In here it's in low concentration, and so the chances of that signal molecule combining, running into that receptor, are then lower. So if we degrade this receptor, if we degrade the signal, this receptor is no longer activated, right? And then that is the stimulus for this G protein to now remove itself from here, right? And then by doing so, right, you remake this GDP bound G protein that is inactive. That's going to find its way back over here and just kind of sit here and waiting for another signal molecule to bind. So that is a G protein linked receptor model. G proteins are varied, but they are also extremely common in many cellular communication models. And we're going to look at a specific example a little bit later in another screencast. Let's look at tyrosine kinase receptors. Again, we're going to have a variation on a theme here. And so in a tyrosine kinase receptor model, we have here the receptor. Now this receptor looks different. It's structurally different. It's still embedded in the membrane here. We still see this embedded in the membrane, but there are two subunits that are separated. Right? There is a binding site for the signal molecule, and there's a binding site for the signal molecule on this one here. There are tyrosines right here. There's tyrosine-rich areas, and again, tyrosine is just an amino acid, so this is our tyrosine kinase receptors, and these are inactive. So we also then have these inactive relay proteins over here that are, of course, inactive. So what gets all this started? Well, the binding of a signal molecule. So we have signal molecules bind. Right? This is kind of step one here. Step two is that we are going to take ATP and we're going to hydrolyze ATP to ADP, lots of it. Right? So this is going to happen 6 times. Right? And we're going to phosphorylate, right, these tyrosine parts of this. That phosphorylation, right, so creates this phosphorylated dimer where the subunits came together. Right? So you're just changing the conformation. You change the conformation when you phosphorylate it. When you add phosphate groups, right? You change the conformation here. And that change in conformation in some way is then activating these relay proteins that over here were previously inactive. And these activated relay proteins, this begins the transduction. This begins the transduction, and so that's what these arrows here. This transduction would then eventually leads to some sort of cellular response. Okay. So again, variation on a theme here, and of course you would get this to become, this tyrosine kinase receptor to become inactive by removing the signal molecules, degrading the signal molecules. So this is another example of receptors. So G proteins, tyrosine kinase receptors. A third model of receptors are ligand-gated ion channels, and we see this in the nervous system quite frequently. But again, variation on a theme. Here's our target cell membrane. It has a protein channel, and then there is a part of that protein channel right there that is the receptor. Right? This is the receptor part that is complementary to the signal molecule. So here's the ligand. It's going to bind. Right? And that binding, it changes the conformation. Right? So this is a theme as well. So binding of the signal, you change the conformation of, in this case, the receptor as well as the channel. Receptor and the channel. Right? And this channel then opens, right? And then you have facilitated diffusion of some sort of ion, right, or other molecule that is not soluble or that is either large or insoluble, right, and will then enter the cell here. Again, you're going to degrade this, right, make it in low concentration. So this is degrade, inactivate the signal molecule in some way, right, to get it to go to low concentration, and then that's going to close the receptor. Right? The last example here is what we'd seen in action in a previous screencast, where now the signal actually enters the cytoplasm. So this signal molecule here, the receptor, is intracellular. Right? So this is an intracellular receptor here. It binds. Here's the activated protein, the activated enzyme now. Right? In this case, this probably happens to be some sort of transcription factor, TC factor. That transcription factor then binds and will induce gene expression, induce transcription, and then translation. And the response here is you get a new protein made by that cell. So you can turn on or off gene expression by using extracellular signals. Okay? So again, this screencast focused on the variation in the receptors and trying to focus here on the G protein-linked receptors, the steps involved with that, as well as the tyrosine kinase receptors. Right? We then also addressed the ligand-gated ion channels as well as intracellular, right, ones. And typically these intracellular ones are responsible for gene expression. That is often associated with the intracellular receptors, is that you're regulating some sort of gene expression. Okay. Thanks for listening, and bring your questions to class.